So today is the 7th week of the webinar program. And today we're going to talk about something really fun and useful. Also something undercover. Undercover means that we don't talk about it a lot, but it's a very powerful parameter. It's called gradient operating parameter. Okay, so today's 7th webinar topic is HPLC gradient parameter, and also a concept called peak capacity. So really two things. And I'm going to actually talk about how do we use them to develop methods for complex separation. I know that we really did not talk about method development yet, which is kind of a big topic, which is coming later on in the second part in farm analysis. How do you develop methods properly? We started to talk about a different way of doing method development, right? So it's going to be a little bit extra for somebody who's more advanced, right? So if you don't know that part of it, don't worry about it. We will talk about it in more details. Okay, I'd like to do something called the forward. I've been telling people that HPLC is not complicated, but it's very complex, right? Now, it's kind of a little confusing, right? People thought complicated means complex. What I mean is that complicated is something that you really need to know a high amount of mathematics, right? In HPLC, you don't really need to know differential equations or engineering processes to understand the process. But it's complex because in order to do HPLC properly, there's a lot of things to worry about. There's a lot of different parts, right? So once again, we have to go back to the goal, right? Why do we do HPLC? So most of the application to provide accurate measurement or quantitation. Quantitation of what? Of key components. Okay, in a sample, most likely using something which is validated, right? So that's kind of the goal for most people. Why do they do HPLC? I've been commenting on what is key component means, right? If you are doing potency, just one thing, then your key component just your API or active pharmaceutical ingredient or the main component. If you're doing a simple mixture, then the two mixture is your component that you're looking for. If you're doing drugs in physiological fluid, right? Now you have billions and trillions of different constituents component in the blood. But we don't care about some of these endogenous components. We only care about the drug and the metabolite. So that makes things much easier, right? So key component is your intended purpose of your assay, right? So you have to know what you're looking for. Sometimes you don't care about the other thing. You only care about one component or a few components. Okay, very briefly, an HPLC method typically contain these parts, right? So you have sample prep, which can be a big thing, right? A sampling, how do you sample the statistically valid sample, how to extract the sample, filtration, enrichment, cleanup, and sample preparation. How do you prepare a sample, your calibration standard, right? So this is can be a big thing. Just in the analysis part of it, you have to decide which mode to use, right? So we talk about ion exchange, reverse phase, which is the most common mode. And then we have to pick a column. A column have many parts too, the dimension, particle size, the bonded phase type, right? So we talk about that later on in columns. And then you have to select your mobile phase, A and B. A is the weak one, B is the strong one. They can have different types. The pH of mobile phase A is important, and so is the concentration of the buffer or the additive. Then we have operating condition, right? So those are the part we focus on today. What is mobile phase B? The starting gradient condition, the final one, which constitutes something called the gradient range. And even more importantly, gradient time, the time you take from the initial to the final gradients. Now, this is to assume that you have a simple one segment gradient, which is in a lot of the cases, you have one segment gradient. But more and more, we do multi-gradient, right? So things can become very complicated, complex. And then you have flow rate and column temperature. Column temperature is kind of like mobile phase temperature. It's what you set the temperature to be. So we talk about all these parameters. And those parameters are underlooked, meaning that people don't appreciate what they can do, right? So I want to give you the ins and outs of how do you effectively use these things to your advantage. And then you have detections, right? So for UV, it's very simple, just the monitoring wavelength. A lot of time we collect all the UV spectrum using a photodiode detector, which is the de facto standard in pharmaceutical analysis. It's not a single wavelength UV. We use a photodiode detector as our standard detector. And then mass spec, right? So mass spec, you have different type, you have different source. Most likely it's going to be ESI or electrospray ionization in 90% of the application. You can collect MILS, as a complete scan range, or a positive or negative ionization, or sometimes both positive and negative ionization, and your mass ranges. So all these things need to be defined. You can see there's a large number of factors which need to be optimized for a complex separation, right? Sometimes it could be very simple, but in most cases, you kind of have to know how to set these parameters, right? So there's a huge number of complex factor in defining what a method is. And in addition, you have something called quantitation, which is controlled by the CDS. CDS is chromatography data system. People spends a lot of time on the CDS, on the computer, to control the acquisition, the calibration, how do you integrate the peaks, and also how do you report the data to give you the actual result, right? So these are not trivial. They take a long time to do it well. So this week, this hour, we're going to talk about HPLC gradient operating parameter, and how do we use them to develop methods for very complex separation. So those are for some of the more advanced attendees. So agenda is talk about what's isocratic, what's gradient. Now, for most of you, you know what is isocratic, what's gradient. Isocratic are very simple. You have a fixed mobile phase. Gradient, you have a changing mobile phase. And also which one to use, right? So we talk about, well, how do you know which one to use, right? This is actually very simple, but we can make it less simple in this case. And then we describe the gradient parameters, such as the range, the initial and final mobile phase B, percentage B in the gradient segment. So we mentioned about single or multiple segment, gradient time. The one in red are really important. Those are the ones you control for a very complex separation. Then you have flow rate, you have column temperature. Now, all these things can be very infinitely, right? They're continuously variable. That makes things simple. Versus something like a column. A column is digital, meaning that once you change a column, you have to change the column completely. You cannot change it a little bit, right? So for optimization, for method development, these ones are much easier to change. A column is very difficult to change. Well, it's not difficult, right? You have to have the new column. But once you change your method is no longer validated, right? So if you have a regulatory assay, which is regulated, which is documented, you cannot change your column. Once you change it, all your validation data, stability data is gone. Okay, you have to start from scratch. Okay, so how do you use that in method development? And then we talk about peak capacity. What is peak capacity? Now, this is a term that most of us does not use, but it's very important. It's actually much more useful than plate counts. Peak capacity. How many peak can you fit in a chromatograph? So isocratically, the mobile phases affects the composition. You can use pre-mixed mobile phases, by using a measuring cylinder, but you really shouldn't do that. You should trust your pump. Pumps are really good and they can blend it, but much better than you can pre-mix it. Isocratic method has a real problem. It will miss any lay eluter, right? And sometimes the column are contaminated. So gradient in a way, even though it's more complex, you always start with a clean column because you go up to the gradient, you wash out your column every time. The separation power is limited. So is, separation power is measured by peak capacity. Typically, isocratic, the peak capacity is about 100. And we define what is peak capacity later on. Isocratic analysis are the retentions defined by the percentage of mole phase B, the flow rate, and the column temperature, right? But the mole phase B is most important. So you define that first. The flow rate actually is pretty mundane because typically we set our flow rate to be 1 mil per minute. But it's really depending on the column inner diameter, right? So you have to recognize that the flow rate must be commensurate or highly dependent on your inner diameter of your column. For most people, they use 4.6 millimeter ID. They really shouldn't be doing that. I think that everyone should use 3 millimeter ID column for analytical assays, right? And I would explain that later on. For column temperature, most people use 30 and 35 degrees, and we explain why that is so. For gradient assay, you increase the solvent strength during the run. Gradient are better because give you a high separation power or peak capacity, give you sharper peaks, and give you better separation to early eluting peak and late eluting peak. Now, recalling that for gradient reverse phase chromatography, in reverse phase chromatography, the retention mode is hydrophobicity of the analyte, right? Meaning that if you have a compound which is very hydrophobic and not water soluble, then your retention time is going to be later. Or for something that's water soluble, you have no retention time or very low, very small retention time. It's a little bit more difficult to develop, but the equipment is getting better. So it's no longer a big deal if you know what you're doing to develop a gradient method, right? So most of the critical assay are gradient methods. And the gradient profile is once again defined by the range, TG, and the flow rate. And we talk about each parameter in some more details. Okay, so talk about gradient operating parameter. So we're going to do, we discuss it in the reverse fashion. First, talk about column temperature. I'll give you some normal range. Typically, it's 30 to 60 degrees. That's the normal range for most analyte. Now, you must thermostat your column because the retention in most chromatography assay are quite dependent on temperature. If you just leave it at room temperature, it's not good. Your temperature is not precise enough. And we are relying on your reproducibility of your retention time to identify your peak, right? So you must thermostat your column. A high temperature is good to decrease viscosity and back pressure, right? But to a certain limit. I think for most folks, it is about 60 degrees. It is a very useful factor for method development, particularly if you have a very complex separation. You have a lot of peaks. Temperature is an easy factor to change. Because you can change it continuously, it doesn't cost you anything, right? It's not like you have to buy a new column. Flow rate is also quite important, right? But you don't really change it that much, right? You have to know what is the right range. Most people use 1 mil per minute for a good reason, right? We talk about why they should use 1 mil per minute. A flow rate is highly dependent on the column ID, right? Because if you have a small column, you have to use small flow rate. If you have a prep column, your flow rate might be 2 gallons per minute, right? So totally a different range. But once you define the column, your flow rate is almost set, right? Within a certain range. Always better to use higher flow rate and gradient. Give you higher peak capacity. and peak resolution. So we explained that later on. Why is that so? Why is high the better? Now, obviously, you have a pressure limit. If you have HPLC, you don't want your pressure to go above 4,500 psi because your pump would shut off at 6,000 psi. You don't want to lift too close to the shutoff. And then today we talk about some of these things: gradient time. Why do we use linear gradient? And why do we want to use multi-segment gradient? Right? So I'll give you some examples. Now, this is a simple gradient profile, right? Most of you do that and realize that, but it's really a powerful method, right? Why do I call it powerful method? It is a simple, broad gradient, right? So I start with 5% B, 5% acetyl nitrile methanol, go all the way up to 100%. Sometimes you go to 95%, right? But you can go to 100%, right? It's almost the same. And then most gradient profile have a purging step. A few minutes, at 100% B. And then you have to equilibrate back to the starting condition, right? If you don't equilibrate back, you will not get reproducible gradient analysis. So equilibration typically is 5 times the column volume, right? So typically about 2 minutes in modern HPLC. And then you ramp back, right? The ramping back is rapid. You can use 0.5 minute. You can use 0 minute. You can use 1 minute. But you do it very quickly, right? You don't want to waste time, right? There's no point to ramp back very slowly, right? Do it very quickly. Okay, so this is a simple broad gradient profile. Now, I also call this method a universal screening method. Now, this is a very powerful method because with this single method, you can almost analyze everything in the world, right? Because most things have different solubility in water, right? Most components in the world. And this reverse phase gradient allow you to have one method that works on anything. So this is very close to a universal method. You can use this type of method for reaction monitoring, if you have organic laboratory. And we do that all the time, right? Use this type of generic gradient to look at our reaction, to monitor it, to make sure that our reaction are completed. Now, obviously, you must separate the product from your starting material, right? So, but a single HPLC gradient in conjunction with UV and mass spec would do that very, very useful in most cases, right? So this is a very powerful. So it is actually a generic method for sample screening and reaction. It's called IPC, in-process monitoring or sample screening. And for purity assay, right? So if you want to analyze the purity of anything, just do a generic gradient like this, and it will work. So this is a really simple example, right? So 5 to 100% B in 10 minutes. It's a nice number. It's a nominal number, 10 minutes. Now, a lot of labs use ballistic gradient, right? Ballistic means it's like a rocket. It's like a rocket that goes very fast, right? So it means that you do 5 to 100% B in about 1 or 2 minutes. It's called a ballistic gradient. And this particular assay is called high throughput screening, or HTC, right? High throughput screening. So once again, you have a very fast assay, 1 or 2 minutes, that works for almost everything, right? So it's a powerful generic method, right? However, if you do it in 1 or 2 minutes, your peak capacity, your separation power is not too good, right? Because you push things out too quickly. So if you want to have more separation power, you slow down the gradient to 2 minutes, to 10 minutes, to 5 minutes, or sometime to 20 minutes, right? So if you want to have a high resolution, you need a long column and a long gradient time. Now, why do we start with 5% B, right? Now, sometimes we can go down as low as 2% B. If you have something that is very water soluble, you have to start low, right? But very rarely do we go to 0% B. okay, you almost never start with water because water have very high surface tension. And there's a phenomenon called phase collapse, or the C18 chain, which is hydrophobic, they are not being wetted. Okay, if you have water, they will not wet C18, right? They are not kind of miscible, right? So you need a little bit of acetonitrile methanol to keep them wetted. If you go to 100% acetonitrile, then the brushes actually would not stick together. They would be floating, kind of, right? So this is a phenomena that people need to kind of pay some attention to. You don't want to go too low, right? So usually we don't go below 2%, except that some columns allow you to go low, right? We talk about that in the column section. Now, what is the total analysis time? Total analysis time is the gradient time, in this case, 10 minutes, plus the purge time, which is 2 minutes. The Rembeck time is another 2 minutes, another 2 minutes of equilibration time, right? So a total analysis time is 16 minutes, right? It's also called the total sample turnaround time. So every time your auto sampler inject a sample, 16 minutes later is ready to inject another sample, right? And then you have to need some time for the auto sampler to clean itself, right? So 16 minutes is kind of the total analysis time, right? So it's not just the gradient time. But the total analysis time is always dependent on the gradient time. The gradient times dictate the analysis time plus some of the purging time. Now here is an interesting proposition, right? When is isocratic analysis more appropriate, right? So it's kind of a stupid question, right? It's kind of a question which is not quite modern, right? So today we don't really ask this kind of question. But anyway, once upon a time, people really want to do isocratic analysis. Or the QC manager demand isocratic analysis because they like to release product. They don't want something that's more complex like gradient, right? So I would say 20 years ago, all the QC tests are isocratic. They just disdain, they don't want to do gradient, right? Yeah. And I have actually an experience with a QC manager who doesn't really like gradient, right? But today you really cannot do that in pharmaceutical analysis because things are much more complex. So here is a study that's being answered by Dr. Snyder. Okay, so they invented a rule called the 0.25 delta TG. TG is the total gradient time, right? So what he said, what Dr. Snyder, who's being called the father of liquid chromatography, said that if you have an assay and all your peaks eluded between 0.25 delta TG, then you can probably do it in the gradient, right? If it's larger than that, you should do a gradient. I'm sorry, if it's smaller than 0.25, you can do isocratic, right? So that's according to Dr. Snyder, right? I don't kind of agree with that, but but this is the rule, which is very simple. Now, what do you mean by 0.25, right? So here is an assay. Okay, actually, it's an experiment that I set up to prove that how that works. So we have a number of peaks in here, right? So 1, 2, 3, 4, 5, 6, 7, 8. We have 6 impurity plus a precursor plus a API, right? Now, if you look at the range, right, it's 5 to 95%, right? So all the peak eluded between 35% of the TG, right? So how do I get that, right? So the TG is 12 minutes. All the peaks elude in about 4 minutes, which is about 35%. So that means that it's exceeded that rule of thumb, right? So you really need a gradient assay to do it properly, right? So you can see that this assay is not perfect because impurity 3 and impurity 2 are called a critical pair, right? They are not separated. It needs some work over there. Now, if we say that, hey, we're not interested in 5 and 6, right? I only interested up to impurity 4, right? This is a theoretical question, right? So in this case, all the peak eluded in about 0.2 of the gradient time, right? So if you look at these, about 2 minutes, about 20% of the gradient time. And then, so that means that you can find an isocratic method, right? So I did find an isocratic method, right? So here's an isocratic method. I forgot what percentage B it is, but it separates everything pretty well, right? So that's what Dr. Lloyd Snyder mean, that you can have an isocratic method, right? But you can also do it in a gradient method, right? Using a shallow gradient, which is my own preference on that. Okay, so if I want to have an isocratic method for everything, right? So here I did try to find an isocratic method. The best isocratic method is about 38% acetonitrile, right? You can see that it's not perfect. Why is it not perfect? Because these two peaks are co-eluding, impurity 1 APIs co-eluding, and also some of the later eluding peaks are just not good, right? They're so broad. The time is 40 minutes, right? So the isocratic method is not good when your range of the analyte is too broad, right? You have too much discrepancy between the hydrophobicity of the components in your sample, right? So I would say in today, gradient is really preferred for all complex samples. Why? Because isocratic have a big problem. You always miss the lay eluder, right? You don't know if you have a lay eluder or not, right? Particularly if you have a purity assay, right? So today, we always do gradient analysis in most cases. And also you clean out the column because very few component would get stuck in the column if you wash your column with 100% acetonitrile. What is peak capacity, right? So just very quickly, peak capacity is the number of peaks that can fit into a chromatogram with a resolution of 1, right? So that's the definition, right? Now here is a gradient, okay? Okay, so in this gradient, the gradient time, did I define the gradient? The gradient time actually about 1.5 or 2 minutes, right? So you can see that all the peaks have a peak width. All the peak width in the linear gradient are very similar. Okay, you take a look at that, they're almost the same, right? Which is totally different than isocratic separation, where the peak width keep getting broader, right? So the peak width in a linear broad gradient is very similar. The peak capacity defined by this equation, the simple equation, it is kind of an estimation. 1 plus the time Tg, assuming a simple broad gradient, divided by the average peak width. And peak width is really should be the same. So you can either take the average peak width or you can just measure one of them. It should be the same, unless one of the components are very basic or something strange. Here's a better or more complicated, complex definition, right? This is developed by Dr. Uwe Neuer. I know Uwe for many years. He was the director of research in waters. He was responsible for manufacturing all the waters column for about 20 years. Now, he happened to be a German engineer. He's a chemical engineer. He knows math really, really well. So he actually worked up the equation, which is a better equation for peak capacity, which is much more complex, right? Most people would just use a simple one, right? You need to know the Van Diemen's B term, the Van Diemen's C term, and you have a TG. Okay, so that he defined a TG a little bit different than mine, right? Okay, the G is a small lowercase, right? So I always use uppercases. Okay, just a little bit about peak capacity. Now, what it is, what are the common values? So isocratic analysis, typically up to 100 peaks. You can separate in a chromatogram under isocratic condition. If you have a gradient HPLC, using a pretty good column, a 3-micron column, 15 centimeters long, peak capacity is up to about 200 peaks. Now, doesn't mean you actually can separate 200 peak baseline, right? It means that it can fit 200 peak. If they are irregular peaks like an oligomer, right? And if you're lucky enough, they behave perfectly, you can line up one by one 200 peak. People complain about HPLC because the peak capacity is not as good as capillary GC. Capillary GC can do 1,000 very easily. However, with UHPLC, ultra high pressure LC, we can get easily 400. And if you use longer time, you can, without a lot of trouble, get to 800. So it's getting close to capillary GC. Now, what if you have a very complex sample? What do you do, right? So you can do what called 2D LC. You can use two-dimensional LC and couple them together in a comprehensive way. Comprehensive means that everything that go inside the first dimension separation go into automatically on the second dimension. The second dimension is very quick, right? So it have to trap each peak and have to do it online very quickly. You can also do it offline by trapping each fraction. So it's called two-dimensional HPLC. Now, if you know what you're doing and the two columns are orthogonal, orthogonal means that they have different selectivity or different separation mode, then your peak capacity is 40,000, right? So you take the product of the peak capacity. Now, why do you need high peak capacity? Now, if you have a problem such as proteomics, right? First of all, if you want higher peak capacity, your HPLC is better because you use small particle, high pressure, is much better solution than for complex sample. Two-dimensional LC or two-dimensional UHPLC is much better for a very complex sample. In proteomics, right? What is proteomics? Proteomics is a study of all the proteins in a living organism. Typically in your body, you have 100,000 proteins. So it's a very complex problem, right? If you can define all the proteins and understand their function, you understand how the organism works, understand how do you prevent disease, how do you mitigate them. So a lot of people are interested in proteomics, but it's a complex problem, right? You have 100,000 proteins, which are codable, are coded by your genomes. And there are also other proteins. There's actually much more number of protein. They are minor proteins. So it's a very complex sample. So to do it properly, how do you do it, right? So a few laboratory uses this type of assay. You couple ion exchange chromatography with reverse phase, and then you go into a mass spec. So right away you have 2-dimension plus mass spec is another separation. You have 3-dimensional separation power, right? The best lab actually would do something like a QTOF, a quadruple, a time of flight, and then coupled to an IMS, ion mobility mass spec. It has a power to separate isomer, right? So this is kind of the best of both worlds, right? But you generate tons of data. If you know what you're doing, you're overwhelmed with data, right? But this is kind of a system for very complex sample. So this is a 5-dimensional nano HPLC plus mass spec. Mass spec is actually a separation technique. It's separate by a mass to charge ratio. And you can do it very quickly in milliseconds, right? It's much quicker than any HPLC. However, you need a high vacuum. It's not perfect. So you still need a separation before that for very complex sample. So here is the scheme in one paper. It's an older paper. Actually, it's developed by folks from Agilent. And it's probably quite obsoleted in proteomics, right? But it shows you the complexity of a multi-dimensional HPLC with mass spec. Okay, easy way to increase peak capacity is to increase retention time, right? So here I did a study myself. This comes from my own lab a few years back, right? So I have this, this is the Agilent test mix. Any surface engineer is familiar with the test mixture, right? So it's 9 alkylphenols. If you use UHPLC, so I use one of the best column, Waters 1.6 SPP, superficially porous material, very small column, 1 mil per minute with this gradient. a gradient time of 1 minute, 3 minutes, of 10 minutes, right? So I actually measure the average peak capacity, right? So I get a peak capacity of 96 for about 100 for a 1 minute gradient. If you use 10-minute gradient, you can go up to a peak capacity of 300. So with a little column, we can do a lot of separation, right? And this column is very quick, right? So if you know what you're doing, you would use UHPLC. A short column, you can do a lot more in very short time, right? So this is the new way to do method development using UHPLC, right? And you can see how powerful by changing the gradient time, right? You can have very fast analysis with good resolution. You can have a longer separation with very good resolution. And the gradient time actually is not too bad, it's 10 minutes, right? Here's an actual example. Here is a generic method that I developed for cleaning verification, right? You need to do cleaning verification under GMP manufacturing to make sure that the vessel are being cleaned, right? So for every project you support, you need to do a cleaning verification, which is a GMP application. So at that time, I was supporting this compound, Genentech C, right? This is an AKT inhibitor. And we were supporting it. I developed a method, a generic method, very mundane separation, right? 1 mil per minute. And the gradient is 10% to 70% B in 7 minutes, right? So nothing unusual. 1 mil per minute. And you can inject a rinse of the vessel, either a tumbler or a reactor. You rinse with methanol, you dilute with water, you inject 20 microliter. And it's a generic method for all these compounds, right? So at that time, once I developed a method, there are 3 projects going on, A, B, C, in the pilot plant at that time. So all 3 compounds can be done by a single generic method, right? So why do we develop another method, right? So just this generic method will work really well. And we get more greedy, right? So we say, why do I put all the portfolio? So at that time, this company actually are supporting about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different small molecule project going on. And they all work really well in this one generic method. So one generic method, you can handle the entire portfolio of the small molecule campaign in this one company, which is substantial. Okay, now the question is, right, so most small molecule drug, we elute before 70% acetyl-nitrile, right? So very few drug actually elute at 100% acetyl-nitrile, right? So the question is why? Why we don't have very hydrophobic drugs? And actually, for some of you actually that understand drug development, right? There's a rule called the Leon Lipinski's Rule of 5. For a drug to be orally available, right? Orally available drug, meaning a drug that can be ingested as a tablet or capsule, right? So you don't want a drug which is too soluble, because then it will not get absorbed by the body. Your colon will not be able to absorb it. It will get excreted. If the compound is too hydrophobic, it will not be bioavailable. It will not be permeable. So most drugs actually would elute before 60 or 70% acetyl-nitrile, right? So when we do IPC, we don't do from to 100%, right? Now you can purge a column to 100%, but your assay is to 5% to 60 or 70% acetyl-nitrile, right? Because most drugs would be eluted before that, right? If it elute to 100%, it would not be a good oral drug. Now, some of you actually see this type of gradient curve, right? Convex and linear and concave curve, right? So a company, Waters actually been advocating for this type of gradient curve, right? Because they can do it, right? The microprocessors have no trouble doing that. But you should not use any gradient curve, which are, they're really cute, right? I'm a convex and concave, right? You should only use linear curve, like gradient curve number 6. Now, why don't we want to use all these nice looking curves? Because once you use these type of curve, it's almost impossible to transfer to a different system, right? For instance, if you want to run your method using a gradient curve, which is nonlinear, and transfer it to Agilent, you cannot do it. Okay, so you have to use Waters system to do that, right? So that is really not good. We want our method to be universally executable by all the different equipment, right? So today we don't really use it, right? So we only use linear curve, and typically from two binary solving, right? So that is the norm. So even though you can do it, doesn't mean you should do it, right? Oh, very quickly, I want to talk about multi-segment gradient. You see these more and more, right? And why are they very common in stability indicating assay of pharmaceutical? So the reason why this is so is because in a pharmaceutical compound, which is stability indicating, most of the problem come around the main peak, which is the API peak, the active pharmaceutical ingredient. Because most of the impurity and degradation product are related to the API. We call it related substances, right? They're related. And in most cases, the degradant, the core of the molecule is not broken, right? Yeah. Occasionally, they might hydrolyze into two components. Most of the drugs today are made from multiple motif, right? You have molecular weight motif. Typically, they have 2 or 3 parts that are coupled together, right? So if the core of the molecule is not broken, in general, they would not be broken. Then the hydrophobicity is very similar, right? And you have all the isomers, diastereomer, and most of the small molecule drugs are also chiral or multi-chiral. So you have a lot going on around the API peak. So you want to have a shallow gradient to maximize the resolution around the API peak, right? So that's why it's more like the norm for a complex drug substance or drug product method to have multi. Now, always you have dimer, so you always need a purging step to get rid of the dimer or to assay for the dimer, right? But you don't need to have very high resolution, right? Because things are much simpler during the purging step. You collect data in the purging steps. Okay, so here is a 3-segment gradient, right? So you have the primary gradient segment, which is a shallow gradient with the API eluting in the middle or close to the end of the gradient, right? Occasionally, you need to have a have an initial gradient step to pick up any hydrolytic degradation product when the API molecule is broken. If you elute at if you just use a shallow gradient, it might not have enough retention for some of the degradation product. And you definitely need a purging step and a equilibration step, right? So here is a multi-segment gradient. The total analysis time is 42 minutes, right? And I show you when I use this gradient profile, right? So I supported, so this 3-segment gradient profile was used to resolve diastereomer around the API, right? So I support this project for about 5 years, right? I took it from clinical step phase 0 all the way to phase 2B. Develop all the methods, develop 40 methods for this project. It is an AKT inhibitors, and it have 3 chiral center, right? So there's a lot happening around the API peak. We have to control the stereochemistry of the drug substance by resolving all 4 pair of diastereomer, right? So we talk about that later on. You can see that it's a 3-step gradient. First step is 5 to 15%. The primary step is 15 to 40% in 25 minutes. You have a purging step, purging gradient of 3 minutes. You hold for 2 minutes, and you equilibrate, right? So total is 42 minutes. This is the primary method. I also developed a secondary orthogonal method to make sure that I have nothing hidden underneath the API peak, right? So we talk about that later on. What is orthogonal method? And that is using a phenyl column with methanol with a different solvent gradient, right? So this gives you better resolution around the API peak. Here is the GLP tox log, right? So it's the good laboratory practices toxicology lot, right, which show you it's very pure. I have a little bit of diastereomers. Here's another example, right? This is one of the most difficult separation. This is for a real drug. It's an HIV standard of care combo drug product. In order to control HIV, you need to take a cocktail of drugs, right? So in the old days, and the leader in HIV drug product is Gilead Science, an antiviral. They actually configure a drug product containing 4 API. Now, the method development of this drug is very, very difficult. And how did they do it? They have 4 API. Each of the API have their related substances with the hydrophobicity around the API. So they actually developed a method using a 5-segment gradient program, using a single buffer and a single mobile phase, right? So this is a really, really nice method. It's not perfect, right? You can see that some of these operations is not baseline, right? But it's definitely accepted by the FDA. They validated the method, and the development and the separation of this drug product is documented in this. If you're interested, that's the references to go to it, published in 2014. Okay, very quickly, flow rate, right? Why is high flow rate good in gradient analysis? Okay, most HPLC method by default use about 1 mil per minute, right? It's kind of a nice round number for flow rate, right? But it doesn't really have to be 1 mil, right? Why 1 mil? 1 mil is a good fit. Most people use a 1 liter solvent bottle, right? And you want to do a 24-hour analysis, right? So 1 mil per minute would be a good number. If you have a leak, you can see it, right? If you do nano LC or micro LC, you cannot do UV detection, right? And if you have a leak, you wouldn't know it, right? So you don't do nano or micro unless you really need to do it. Now, it happened that 1 mil per minute is also the optimum flow rate, which we would talk about later. For 5 micron column with an ID of 4.6 millimeter ID, right? So this should be 4.6, sorry, I should fix that. So this is the standard column in a diameter for many years, right? I think it's too big. I think people should use 3 millimeter ID column, right? And nobody should use 5 micron in today's HPLC, right? You really should use 3 or sub 3, or even sometime sub 2. So the flow rate should be proportional to the square of the inner diameter of the column, okay, or the radius of the column. And the optimum flow rate also somewhat dependent on the particle size, right? So this optimum flow rate that gives you the best efficiency, there's a range that you should use. The reason why high flow rates are advantageous is because it can reduce analysis time. So if I show you that example, right? If you use high flow, you can use less gradient time, give you equivalent a gradient volume, something that we call it. You can pump as much volume through it in less time. So the peak capacity is the estimate of peak capacity is actually proportional to the square root of the n, the plate count, or the square root of the gradient time, right? So this means that a longer gradient time is good for peak capacity of give you overall peak resolution. We can define something called gradient volume, right? Which is gradient time by the volume, by the F, right? So if you use a higher flow rate, you can reduce the gradient time, right? Which is good because then you can go home early. You have a quicker analysis time, right? So I show you one example, right? So here's the example. If I use, remember we showed an example of a 10-minute gradient with a total gradient time of 16 minutes, right? I can do the same gradient, have the same peak capacity. If I use 2 mil per minute, I can do it in 8 minutes, right? Because then my gradient time would be 5 minutes, right? So it's equivalent because the gradient volume, which is the gradient time time the flow rate, is actually equal to 10 mil, which is the same as the 10-minute gradient, right? Okay, I show you an example for high throughput screening. It's a real example. This is a case study in high throughput screening. It's a lab in Genentech, which support drug discovery. Now they do thousands of drug candidate a year, right? So they actually cannot use a customized method, right? They use a generic HPLC UV mass spec method, right? So if you're interested in that assay, this is the publication in 2015, right? So the first author is Mingling Wang. She is the manager for the support laboratory. She did this job for many years. Okay, so typically they use a column, which is this column, a very short column with very small ID. It's a very small column, packed with 1.7 micron, right? 35 centigrade degree, right? The void volume of this column is 0.2 mil. It's a very small column. And the generic gradient are acid and base, right? Now in this case study, we did a case study of using 4 drugs, right? So, which is unusual, right? So we have 4 different drugs, and the basic pH mobile phase A using 0.1% ammonia actually works better because the formic acid give you a co-elution between B and P, right? B is this compound, and P is propranolol. Okay, so here is the for each individual injection of the single drugs, right? So here is a case study that we did, right? So in 1.5 minutes, the gradient time is 1.5 minutes. A full gradient, you can have pretty nice separation, right? Now, why do we call it high throughput screening, right? Because in 1.5 minutes, which is not a long time, the gradient volume at 0.8 mil per minute, which is pretty high flow rate for that column, is 1.2 mil, right? But 1.2 mil is 6 times the column void volume, right? So you actually wash the column with 6 times the column, 6 times the void volume of the column, 6 times, with increasing amount of acetonitrile in this column, right? So the gradient did most of the work, right, in this high throughput screen. You're pushing out the compound using the gradient, right, rather than just the efficiency of the column. The column is actually pretty efficient. I probably have about quite a bit of plate count, right? So so this is high throughput screen using a small column, a high flow rate. Now this flow rate doesn't look too high, right? But if you translate this flow rate for to a 4.6 moment ID column, right? It's equivalent to 4 mil per minute on a standard ID column, 4.6 mil ID, because we are using a very small ID column, right? So all these things are complex, right? Because they all depending on a large number of factors. A column temperature is important, right? We always control the temperature. The default temperature is 30 or 35 degrees centigrade. It is not room temperature, right? Because at room temperature, you have no temperature control, and you don't have very precise retention time. Okay, the reason why we do this is because most column oven, they might not have a Peltier cooling device, meaning that you have to do room temperature plus 5 degrees, right? So that's why 30 and 35 is the default. Typically, sometimes we use high temperature, but very rarely we use it above 60 degrees, right? Occasionally, we would go up to 80 degrees, right? And we use high temperature for protein and peptides because we get better peak shape. We have higher protein recovery using higher temperature, right? This is specifically for proteins and peptides. Okay, so retention time is highly dependent on temperature, particularly in reverse phase chromatography. Is less dependent on ion exchange. The equation controlling the retention of reverse phase is defined by the Van Hoff equation, right? This is a famous equation in physical chemistry, where the log of the K of retention is controlled by this equation, right? T is your temperature in Kelvin, right? It's not in centigrade, right? So temperature is optimized also in complex separation, since often temperature affects selectivity to some small degree. If you use dry lab, if you use a simulation program called dry lab, the standard parameter you fix that you try to vary to generate that simulation is gradient time versus and also temperature, right? So temperature is, even though it's not very, it's a big factor, it is variable very easily, right? So something that you can use for complex separation. A low temperature sometimes is helpful, particularly in isomer separation. One case in point is separation of rotamers. Rotamers are cis and trans isomer with a restricted rotation, right? So sometimes if you go to lower temperature, you can separate the two rotamers, cis and trans. While if you use high temperature, they would interconvert, right, on the column during your analysis, right? So you have to low temperature is better. So summary, isocratic separation are used for rough potency or dissolution testing or simple mixture, right? So a lot of the older QC method, including most of the USP method, are using isocratic analysis. And most of them don't work really particularly well, right? For purity assay, you really have to do a gradient. A gradient method I prefer because of higher peak capacity and also better mass balance, right? Meaning that you have a clean column to start with. You can get all the lay eluders out. It is really a mandatory for purity testing or stability indicating assay. Important parameters are the gradient segments, either single or multiple gradients. Gradient time are very important. Flow rate and temperature. In general, higher gradient time plus flow rate use higher peak capacity, and in general give you better resolution. Peak capacity is defined by this equation, basically proportional to the gradient time divided by the peak width. It's the number of peak that can be separated in a single linear broad gradient. Okay, so here's the references.